Root knot nematode is currently one of the major pests and diseases in greenhouse vegetable production, which seriously affects the yield and quality of greenhouse vegetables. How do we present and control root knot nematodes? Today, I will introduce to you what root knot nematode is, as well as its pathogenesis, damage symptoms, development pattern, prevention, and control methods. Next, let's come to the details. First, let's look at what root knot nematode is. Root knot nematode is a highly specialized, omnivorous plant, pathogenic nematode that is widely distributed throughout the world and has a wide host range according to incomplete statistics. Root knot nematode hosts have exceeded 3,000 plant species, including food, economic, vegetable, fruit and flower crops, with Solanaceae, Cucurbitaceae and Cruciferae being particularly serious. Its harm has exceeded that of bacteria and viruses, and it is the second largest plant disease after fungi. Once it occurs, the yield reduction is 10% to 15% in light cases and as high as 75% in serious cases. It is also one of the most important causative factors for inducing diseases in plants, according to the International Root Knot Nematode Organization. There are 4 commonly occurring root knot nematodes: southern root knot nematode, Javanese root knot nematode, peanut root knot nematode, and northern root knot nematode. They cause more than 90% of the overall crop losses caused by root knot nematodes, of which the southern root knot nematode is the most damaging and is a priority for control. Second, pathogenesis of southern root knot nematode. The southern root knot nematode is a specialized parasitoid mainly on the fibrous roots or lateral roots of the roots, and harms the host in 3 ways. 1. There is a direct epidermal cell, which destroys the epidermal cells of the host. 2. It induces other pathogens to infest and harm the plant. The mechanical damage produced by root knot nematodes to the plant provides a convenient channel for the infestation of soil-borne pathogens such as wilt and root rot fungi. 3. It pricks the host with mouth needles and secrete poisonous substances which contain a variety of digestive enzymes, inducing various pathological changes in the host tissues, destroying the normal metabolic function of the host cells and producing lesions, causing deformation of the root surface or damaging the internal tissues of the plant, and then forming candida-like rhizomas of varying sizes. The rhizomas are initially white and soft in texture, later becoming light brown with a cracked surface. Third, the symptoms of root knot nematode damage. The most typical symptom of the southern root knot nematode after it has damaged a plant is the giant cells that are formed, and as a result, on the roots, tumors of varying sizes called root knots can form. After the occurrence of root knot nematodes, the ability of the root system to absorb and transport nutrients and water decreases, forming weak seedlings and affecting yield. The symptoms of light disease. Plants are not obvious. The above-ground part of the heavily diseased plants shows that the plants are of different sizes, stunted, most of them, are relatively short, thin, slow-growing, and often show wilting at noon, recovering in the morning and evening. In severe cases, the entire plant dies outright. The leaves are small, pale, and yellowish in color, look as if they are lacking in elements, and they drop flowers and fruit, which are small and misshapen. Fourth, the pattern of root knot nematode development. Root knot nematodes are colorless and transparent and are divided into 3 main stages: egg, larva, and adult. Of these, the second instar larvae damage the roots. Root knot nematodes are mainly found in the 0 to 25 cm layer of soil, with the highest distribution in the 3 to 15 cm layer. Root knot nematodes often overwinter in the soil as eggs or second instar larvae residues, with diseased soil, diseased plants, and irrigation water as their main means of transmission. The onset of the disease is more frequent on acidic and sandy soils, and soil moisture in the field is an important condition affecting hatching. Generally, the temperature and humidity of the protected area is suitable for the growth of vegetables, but also suitable for root knot nematode activity, which is conducive to the development of the disease, and the disease is heavy in successive crop planting, and the suitable temperature for infestation is 25 to 30 degrees Celsius. Nematodes typically survive in the soil for 1 to 3 years. Root knot nematodes are extremely fertile, usually laying 1,500 to 3,000 eggs per female, and their life history consists of eggs, 4 larval stages, and 1 adult stage. The time required to complete the life history cycle is temperature dependent, typically 25 to 45 days during the crop growing season, and 0 to 5 degrees Celsius kills southern root knot nematodes but not eggs. Root knot nematodes die within 55 degrees Celsius. Fifth, the root knot nematode control methods. Currently, root knot nematode control methods mainly include 1. Strengthen phytosanitary, strictly prohibit the transfer of diseased seedlings from diseased areas. 2. Agricultural control. 1. Select root knot nematode resistant varieties. 2. Seedling soil with grass ash and seedling substrate with 1:1 ratio can control root knot nematode. 3. Planting onions, garlic, and leeks for more than 2 years in plots with serious nematode damage can reduce the nematode base, and planting fast-growing vegetables such as oilseed rape, spinach, and chard to trap nematodes can also reduce the nematode base. 4. Spreading grass ash in the greenhouse at the rate of 100 kilograms per mu, then turning it over deeply can control the occurrence of root knot nematodes. 3. Physical control with high temperature smothering, flooding, treatment, and lime nitrogen method. 4. Biological control, using light purple mimicry and others to control. 5. Chemical control. Chemical control is the mainstay of current pest control. Highly efficient and low toxicity products are recommended for the control of root knot nematodes, such as avermectin cachenti granules and phosphothioate granules. The granules are used at a dosage of 1.5 kg per acre before planting use. Uniform spreading, furrow application. We can also spray cucumber seedlings with 1.8% avermectin emulsion 3,000 times at the seedling stage. At the time of planting, each plant can be used 200 milliliters of this concentration of solution for root irrigation. 3 to 5 drops by drip irrigation during the growing season can be very effective in controlling root knot nematodes. 6. If a soil fumigant such as methampsodium is used, it will kill both the nematodes and the soil pathogens. The application method is as follows. Firstly, till the soil, then 15 to 20 days before vegetable transplanting, apply different doses of the agent evenly on the ground surface, then mix it with 0 to 10 centimeters of soil on the surface and water, mulch for 2 weeks and then remove the film, till the soil, and transplant 2 days later. Today, I have introduced to you root knot nematode control techniques from the aspects of pathogenesis, damage symptoms, development pattern, and control methods. Did you learn it? That's the end of today's lesson. See you next time.